feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. And I'm right where I belong in my happy place. And I'm right where I belong in my happy place.
Hi, and welcome to the National University of Singapore Yong Lu Lin School of Medicine Healthy Longevity webinar. Thanks for joining us again. Excuse me. I want to remind you to use the Q&A function to send questions tonight. We always like your questions, so ask as many as you can, and we'll get them to Tony. Our speaker, as I just mentioned, is Tony Wyss-Coray, so it's going to be an exciting talk about the benefits of young blood. But before we get to him, we have Dr. Wei-Lan Wang, who's a senior research fellow at NUS, and she'll be talking about her lab's project, the use of potential gerotherapeutic drugs and mortality in geriatric rehabilitation in patients.
Thank you for introduction. As mentioned, I'm Dr. Wei-Lan Wang, and today I'm going to share one of my recent work on the use of potential gerotherapeutic drugs and mortality in geriatric rehabilitation in patients. So firstly, let's have a look on why we use repurposed drug. This has been used in many fields such as oncology. Compared with developing a new drug from scratch, it usually has lower cost, and also because these drugs have been used for many years in markets, thus we have a better safety profile. And covers evidence on how we can use repurposed drug as a gerotherapy. This is a review summarize some of the potential drugs as we can see here. But I would like to call for attention that we are still lack of some evidence on the human mortality. As I highlight here, many of these drugs still lack of evidence on this, which is also the focus of today's study. Before we go to results, let's have a look on who are these geriatric rehabilitation in patients. Most of them experience acute events, and many of them have multiple diseases and have long-term medication. And polypharmacy, which means that they are taking more than one drug, is very common in this population. This study is conducted in RESORT, which is a geriatric rehabilitation in patients cohort, coming from the Royal Melbourne University and the University of Melbourne. These cohorts were collected from 2017 to 2020. And the patients have been followed up up to 1 year. And we also collected the list of medications that are used from the comprehensive geriatric assessment and also their medical records. And here is a characteristics presentation for this cohort. Among the nearly 2,000 patients, 1,890 of them, we can see that the average age is quite old, over 80 years old. And after 1 year from discharge, around 20% of them were unfortunately passed away. I would like to call for attention on the CCI score, which stands for the Charlson Comorbidity Index. This is the index assess their comorbidity. The median value is 2, which means that among these cohorts, most of them have at least 2 chronic conditions or 1 severe chronic disease. How about the use of these potential gerotherapeutic drugs? As we can see that the most commonly used one is the ACE inhibitor or ARBs. This is a drug commonly used for hypertension, followed by aspirin beta blockers, metformin and bisphosphonates. Some of the drugs, for example, SGLT2 inhibitors and rapamycin, they are very interesting, but they are not very commonly used in this cohort. Thus, to ensure a sufficient statistical power, only the drugs were taken by 5%, at least 5% in these cohorts were used in this following analysis. Let's come to the survival analysis. As we can see that compared to patients who were not using any of these 5 drugs, the patients who were using the ACE inhibitor, beta blocker, and aspirin, as illustrated in the green line, have a better survival, which means that compared with patients who were not using any, they can have a better survival after discharge. And these are statistically significant. If we look at the metformin, the effect size is even larger, as we can see that is a quite good separation from the patients who were using none. But for patients who were using bisphosphonates, there's no statistically significant difference from patients who were using any. How about drug combinations, considering that many of these patients are taking more than one of these drugs? In the patients who were using 3 or more of them, as illustrated in the purple line, they are significantly higher, which means that significantly better survival compared with patients who were using none. Patients who were using 1 or 2 of these drugs, as we can see here, they are not statistically significant, even though they may show a trend in a better survival. So in summary, the use of metformin, ACE inhibitor or ARBs, aspirin, and beta blockers showed a significant protective effects from mortality. The use of 3 of these drugs showed a higher protective effects than patients who are not using any or only 1 or 2 of these. Combined therapy may offer a better benefit than a single drug. However, I will also highlight that the outcome from this study is completely from an observational study, thus we cannot conclude the causal effects. Clinical trials are warranted to see whether it is these drugs that are really causing a better survival or, let's say, a lower mortality risk. Thank you for your attention.
Thank you, Weilan. Really interesting data. I want to now turn to Professor Tony Wyss-Coray, who's a distinguished professor of neurology and neurological sciences and director of the Phil and Penny Knight Initiative for Brain Resilience at Stanford University. His research focuses on brain aging and neurodegeneration, particularly age-related cognitive decline and Alzheimer's disease. He's received many accolades. I think maybe just one to point out would be that he was named Time Magazine one of the most 50 influential people transforming healthcare. So with that award, Tony, no pressure on this talk. The title of his talk will be Young Blood for Old Brains.
Thank you very much, Brian. Yeah, I'm really excited to be here, and thank you for hosting me. So my lab is really interested in trying to understand how our brains, as we get older, become susceptible to cognitive dysfunction, neurodegeneration, and diseases such as Alzheimer's. And as part of this, we really like to understand the communication between the brain and the rest of the body. So traditionally, neuroscientists have largely focused on the brain as sort of an isolated organ from the immune system, for example. But we know many examples where we have crosstalk between immune responses and the brain, how it functions. Let's say when you're sick, when you get infected with the flu or things like that. Other examples are the gut-brain axis, as well as exercise, which we know has strong effects on the brain, and is presumably through factors that are generated in the periphery, in the muscle, but potentially also in other tissues. And then we have equal evidence that the brain regulates many aspects of body function, and I just list a few here. So we can then ask, in this communication between the brain and the rest of the organism, in simple terms, what goes in, what type of molecules and cells enter the brain, and what cells and molecules leave the brain, what comes out. And we think that this can help us to understand brain physiology with age and disease, potentially exploit transport mechanisms for therapeutic use, but also discover biomarkers and really understand individual variation. And we have used sort of the blood to be at the core of this communication. The blood, of course, connects all the different tissues and cells in our body. It transports immune cells, red blood cells, but it also transports tens of thousands of different molecules. And because we found that the composition of the blood changes with age, as you'll see in a minute, together with Tom Rando, who was interested in stem cell aging in the muscle, we explored whether simply changing the composition of the blood in an adult organism would have an effect on the aging of that tissue. And we were able to show that if you transfer blood from a young mouse into an old mouse, that has profound effects on the brain, including activation of stem cells and generation of new neurons. It increases the connectivity, so-called synaptic plasticity in the brain. So the brain is more able to exchange molecules and connections between different regions. We also get less inflammation, and maybe most importantly, we can improve memory function in these animals. We were also able to show more recently that you can collect the fluid in the brain, the so-called cerebrospinal fluid, from young mice and then infuse it into old mice and get similar benefits as the ones listed here. Particularly, we have also found effects on the cells that myelinate the connections between neurons, the insulation on axons, if you will. And again, we saw improvement in memory function. And then importantly, the opposite is true as well. So if you took blood from an old mouse and infuse it into a young mouse, you can actually accelerate aspects of aging. And this has been reproduced since by many, many different labs. And most importantly, by using completely unbiased approaches. So if you used one of these models that allows you to exchange blood between young and old animals, and then you look with gene expression analysis in an unbiased way at every cell type and organ in the body, by and large, what you see is that the effects are cell and tissue specific. They're particularly prominent effect on stem cells, on hepatocytes and endothelial cells. We can affect aging hallmarks, reduce inflammation by exposure to young, and again, generate the opposite effects if we expose a young organism to old. And people have really reported improvements of function in multiple different tissues, suggesting or demonstrating that there are factors in the blood that can slow down or reverse aspects of aging in the young into old paradigm, or there are factors that can accelerate aging when you put old plasma into young individuals. So we then asked, what are these factors? And can we use the study of blood really to learn about basic principles of aging, the effects of gene, environment, and lifestyle? And if you think about this, the blood, as I mentioned earlier, collects really molecular information from every cell and tissue in our body. And some of these are through active secretory processes, but increasingly we find that cells can release content through exosomes and potentially through other mechanisms in a way that almost every molecule that a cell can produce can end up in the extracellular space, including cytosolic factors, cytosolic proteins, nucleic acid, RNAs, DNA, as well as factors such as transcription factors or kinases. So we can then ask specifically to the brain, are there proteins in the blood that are derived from the brain, and measure with tools now that allow us to quantify thousands of different proteins. Are some of these proteins that we can measure potentially derived from the brain or from any other tissue in the body? And this is something that we have been doing in standard clinical care, in clinical chemistry for decades, but we only measure a few dozen molecules, and most of them are actually not proteins. I listed here from Wikipedia common analytes that are measured in blood when you see a general practitioner and they take a blood sample from you. Only a few of these are proteins, but we measure, for example, albumin and globulins for assessment of liver function, as well as we measure also transaminases and alkaline phosphatase. For the heart, we measure troponin, myoglobin, FABP, a few other ones, and so forth. So this allows us to get information about tissues, and usually we use this to indicate dysfunction of tissue or if somebody is sick. But as you will see, I think we can use this really to look at the physiological state, the health state of a tissue or even of a cell. And so this is an experiment that we carried out a few years ago now, but basically we obtained from the longevity, from the interval study in the UK, we obtained proteomic data for 3,000 proteins from over 4,000 healthy people, age 18 to 95. And so we can look at these proteins. For example, here, a protein that goes up with aging, and we use blue if the level is low and yellow if it increases, if it's a high level. So this protein would be blue in young people, and then it becomes yellow here in this illustration. You could have the opposite effect in a protein that is high when you're young but decreases. Some proteins may be unchanged and others go up and down again with age. And we really find all kinds of different trajectories. And if we look at these 4,300 people, this is what you see. So here from left to right, you have these 4,300 people aligned from 20 to almost 100 years of age. And for each person, we measure 3,000 proteins. And you can appreciate that there are dramatic changes in the composition of the blood as we get older. And it also is important to note that these changes really start from an earliest age. So you're not saved from aging until you're 60 or 70. But what we see is actually that there are dramatic changes occurring particularly at this early stage here, when we quantify the changes that occur at each age, we see very prominent changes around 35 to 40 years of age. We call these waves of aging. Another more subtle wave at around 60, and then the most prominent one in people around age 85. So these are what we can call inflection points, where a lot of things seem to change that lead to changes in the composition of proteins in the blood. We can ask, are there common trajectories, as I showed you before, a protein that goes down with age or goes up with age, and then you have these more complex patterns. So we can ask, using a cluster-based analysis, are there common trajectories of proteins? And we find here, for example, a cluster of 200 proteins that goes down with age and then increases exponentially in very old people. Here you have a cluster of proteins that sort of continuously goes up, but accelerates to increase, and so forth. And we can now ask, what are the proteins doing that have this specific behavior, for example? And what we find is that some of these proteins are specifically derived from particular organs or even cell types. So for example, here, cluster 12 is made up of proteins that are mostly produced by red blood cells and other immune cells. Cluster 33 is made up of proteins that are largely produced in gut tissue, or this cluster here, 26, is made up of proteins that are derived from the brain. What we can also do, because there are these changes that occur with age, we can take any sample, and you will appreciate immediately if you took this sample here that I highlight in red, and you try to fit it in, where does it fit in these 100 years of samples? Almost like a piece of puzzle, it will only fit in here. So what that allows you to do is, if you use machine learning, you can actually generate a model that tells you how old a given sample is, a blood sample that you derive or obtain from a person. So like this sample would be 35 years of age. And it's probably also evident that you may not need 3,000 proteins to make that prediction or that estimate of the age, but you need only a few, often a dozen or so proteins to make that call. What's really interesting is that the proteins that make up this call, so let's say we need 20 proteins to estimate how old a person is. These proteins, naturally, because they can estimate the age, contain biological information about the age. Some of these proteins may just be a reflection of the aging process, but some of them may also actively participate and modulate aging. And we know this because I showed you earlier, factors in the blood can actually change the age of an organism. So it's likely that in any one of these signatures that we call them, you will have a combination of proteins that are causative or are just reflecting changes in aging. So we can then derive models that not only estimate the age, the overall age of a person, but actually estimate the age of each organ. So if you find proteins in the blood that are derived from the brain, and these proteins change with age, you are able to again make a model that tells you how old the brain of that individual is. So this is just exemplified here, where you have the actual age of people. And if you have hundreds or even thousands of individuals, you can try to develop a model that estimates the age of a person based on proteins from the brain. And what you find is, for the majority of people, the overlap between the actual age, the chronological calendar age, is very similar by the age that the model generates. But you will find individuals where you predict them to have a younger brain or an older brain. And so again, the distribution in the population is such that for most people, they fall very close to this line here. And you have this distribution, but then you also find people at the extreme tail ends. And these are particularly interesting. We call them extreme agers. And what's really exciting is that these models, and especially the deviation from your actual age, what we call an age gap, is related to organ disease or future organ disease and mortality. And we did this first, Hamilton O and Jared Rutledge, two graduate students in the lab, in a group of 5,500 individuals. These were in part from memory and aging studies, older people above age 50 or so, where we measured roughly 5,000 proteins, again, with one particular platform from a company called Neurologic. And we built this model for all major organs. And I just show you here an example of a heart ager. So this person, and we found about 2% in these 5,000 people, who have hearts that are clearly much older than the average population's hearts. And when we look at the top and bottom quarters of individuals with young hearts or older hearts compared to the median, you see that over 15 years, so if we take the sample here and we make this prediction of how old the heart is, those with young hearts show very few changes over the age and incidence of heart disease. But if we estimate your heart is older, and again in the top 25 percentile, these have an almost 2.9-fold increase in the risk to have atrial fibrillation or heart attacks. And we can do this for many other tissues. But in the meantime, we were able to analyze a much larger data set from the UK Biobank with 50,000 people. And here, a consortium of companies measured 3,000 proteins with a different platform from a company called Olink. And here we have again the sample at time 0, and then we have follow-up on these individuals over 15 to 20 years now. And what we see is that people who are, when we model their age, the age of their brains, they have an increased risk for Alzheimer's disease. If they have an older heart, they have increased risk for atrial fibrillation. Those with older kidneys, intestine, and immune system tend to develop type 2 diabetes, heart failure, again, the heart plus multiple other organs. And then those with many different organs, clearly older, they tend to have develop chronic kidney disease. And here I just show you the data for Alzheimer's disease. So if you look at all these ages in living people and ask in 15 years, what is the biggest predictor of the development of Alzheimer's disease? We see an 80% or 1.8-fold increase in the risk to develop Alzheimer's disease in those with older brains, and then some of the other organs contribute as well. But if we look now at the extreme agers, so we look at those with 1.5 standard deviation higher or lower age gaps than the overall population, so people with the youngest brains compared to the people with the oldest brains, now we see actually an 18-fold difference in the risk to develop Alzheimer's disease between these extremes, or an 80% reduction if you have a very young brain from the median, so a very strong protective effect to develop Alzheimer's disease 15 years later, or a 3.4-fold increased risk to develop Alzheimer's disease. So again, the difference here in this analysis is that we look at the extreme ages compared to here, where we look at all individuals who have an older age gap. What are the proteins that are made up of this protein of the brain signature? It's only about a dozen proteins or so. They are predicted to be synthesized in the brain, mostly in brain cells. And together, the signature suggests that there is something abnormal with the extracellular matrix, specifically the matrix around synapses, so the connections between neurons. And in one particular aspect, they're called perineuronal nets. They're like a basket that wraps around of extracellular matrix proteins that wraps around the synapses and has sort of holes where other synapses can then make contact and really facilitate signaling between cells. So this is an extremely interesting observation that is consistent with latest discoveries that, first of all, in Alzheimer's disease and cognitive impairment, the earliest changes seem to be of synaptic origin, synaptic changes, and that potentially changes in the extracellular matrix are at the core of these abnormalities. And we can now use this to better understand biology, but potentially then, of course, also to make predictions about the likelihood of somebody developing cognitive impairment. We can also use this knowledge to try to predict mortality. So how likely is a person to die in 15 years? And what we found here, to our surprise, is of all the organs, the age gap, and again, we look at the extreme age gaps, the individuals with the oldest brains are the most likely to die. If you have multiple organs older, there is a continuous acceleration or additive effects of risk to die with odds ratio of up to 8-fold. So an 8-fold increased risk to die. And then equally exciting and surprising, the strongest effects for longevity or reduced mortality is to have a young brain or a young immune system. And again, there's an additive effect here. And if we look at survival curves, So here at time 0 is when we obtain the blood samples. We estimate the age of each of these organs. And so we look at the effects of extreme organ age. In gray is the average population, so no extreme organ ages. If you have a young brain or immune system, you have a slightly reduced mortality. If you have 2 to 4 organs older, this is the mortality curve here, and you can see the more organs are extremely old, the more likely you're going to die. With 8+ organs, about half the individuals have passed away after 7 years. So a very strong predictor of mortality that again suggests that we can capture biological information simply by looking at blood proteins that are derived from different organs. We can also look at self-reported data. This is not the strongest data because this is sort of based on recall. You ask people, did they smoke? Did they drink excessive alcohol? Sometimes it's also not accurate what people record. But by and large, what we find is that people who smoke, they have older organs, shown here in red. Excessive alcohol is associated with older organs. Deprivation index, so socioeconomic status, living in poverty is associated with older organs. And then consumption of poultry, oily fish, vigorous exercise and education tend to be associated with younger organs. Maybe a bit more certain data come from on this right-hand side here from individuals, postmenopausal women who take estrogen replacement drugs such as Premarin. And here we find, consistent with the literature, that many of the organs actually benefit from these drugs, and we see younger organs, except the heart. And again, that's consistent with some of the studies out there, that the heart may be slightly more susceptible to disease in the future. Glucosamine also seemed to have a positive effect as a supplement that a lot of people take. And we have actually found this association even in the smaller cohort of 5,000 people, but we didn't report it because we didn't trust the data, because we felt in 5,000 people maybe this is just an artifact, but clearly reproduced here in 50,000 people, suggesting this has a beneficial effect on overall aging of organs. Vadim Gladyshev recently analyzed these same data, and he has a very strong conclusion suggesting that the age of an organ, the predicted age of an organ, is really an indicator of whether you will get disease of that organ in the future. So in other words, if you can estimate that you have an older kidney, you will get kidney disease later on. This is a very strong statement. I think we will probably need additional data to really validate that. And then many organs, of course, there are different forms of disease. So we have a very good prediction for brain age and Alzheimer's disease, but brain age and Parkinson's is not a good correlate. So an old brain is much more susceptible to Alzheimer's than to Parkinson's, it seems. So we will need more resolution, I think. And this is what we're trying to do with something that we haven't published yet, but basically build models for cells. So by the same argument that you find proteins that are derived from an organ, I showed you earlier that some of these signatures that we see, these clusters of proteins, they're actually localized to individual cell types. So we were able to build, based on the protein measurements that we had available in another cohort here from 1,400 healthy people with 7,000 proteins, we built models for predicting the age of almost 50 different cell types here. For example, liver cells, smooth muscle cells, monocytes, macrophages, oligodendrocytes, and so forth. And you see, again, we don't need that many proteins. So these are proteins that are preferentially produced only in one cell type. But if you add up all the different proteins that we use in these models, we usually get a very high confidence that together this signature is a signature of that cell type. And what's really exciting, this is a cohort called the Global Neurodegeneration Proteomics Consortium from Gates Ventures and Janssen Pharmaceuticals, which will become available to the public in early July. It's basically a collection of about 35,000 individuals with who are healthy or have different neurodegenerative diseases listed here. And we use this cohort to estimate whether these individuals with different diseases have aged cell types. And what stands out immediately, and this is the most striking finding, is that people with ALS, so this is a muscle disease, they have very strongly significant aging of skeletal myocytes as well as cardiomyocytes. This is something that most people have not really looked into, because this is always considered a motor neuron disease. But of course, motor neurons activate these muscle cells and innervate the muscle to induce its activity. I found a few reports that suggest that people with ALS have actually smaller hearts. There's shrinkage of the heart tissue compared to healthy individuals. And some of them, very few, but still a significant number, have sudden heart failure. And so maybe this is something we pick up here with these aged cardiomyocytes. There's many more data that are interesting, and I don't have time to get into, but you see in Alzheimer's disease aging of many different cell types. But it was exciting to see that oligodendrocytes, as well as excitatory neurons, seem to be some of the most prominently aging cell types in these individuals. So I want to conclude by just telling you about our attempts to determine whether a protein really originates from a specific cell type, such as from a neuron, when we measure it in the blood. And for that, we use, this is work from Ian Goldner, who is now an instructor actually at Stanford. We use a technique that is called BondCat, which allows you to incorporate an amino acid that has a chemical modification into the growing polypeptide chain. We made actually a new model here that incorporates phenylalanine that has an azide, an acido group. When you produce now a protein, some of the phenylalanines have this chemical reactive group, and we can derivatize them and isolate them, and then measure them by mass spec and identify what type of a protein this is. So by putting this system, this machinery, this labeling machinery into neurons, Ian was able to label neurons of these mice, and you can see here we get a lot of labeling of many different proteins of different sizes. By histology, we can show that neurons are labeled with this system. By then doing mass spec, we can show that essentially all parts of neurons are labeled, from the synapse to axons, the cell body. We can really capture proteins from all these different subcellular compartments of neurons. And again, we label these proteins in the living mouse. So we can label for a day or for a week, and then we can stop labeling and we can look at protein turnover. We can look, are these proteins ending up in other cells? We see them, for example, in microglia, which are the macrophages of cells. We also see different proteomes in different brain regions of these mice. But really what I want to show you here, what's relevant here, is if we detect a protein that has the label, that has a tag in the blood, we know it must come from a neuron, because only neurons are able to label proteins. So we use this to basically prove that, at least in the mouse, proteins that we measure and that have this tag are from neurons. And so they allow us to record physiological function of neurons potentially, but clearly something that the neurons produce and ends up in the circulation. And we were really excited to see with the highest sensitivity of mass spectrometry in collaboration with Alma Landberg, that we can detect over 600 proteins in the blood that are tagged. So these are all proteins that are derived from neurons and are now in the blood circulation. And excitingly, about 100 of those, so 20% roughly, are derived from synapses. And synapses are really the key. I mentioned to you that they degenerate. They show degenerative changes from very early adulthood until continue to degenerate and disappear as we get older. And they're probably one of the key drivers of neurodegeneration in Alzheimer's disease. And by being able to measure so many of these synaptic proteins, we think we might be able to get really much more information at a molecular level about the cellular state of different types of neurons even. So to summarize, we find plasma protein changes with age provide insight into global aging patterns, relative age and physiological state of cells and organs. Protein changes reflect physiological state, but also are drivers of that state, as shown by the rejuvenating effects. And then we find these signatures of extreme organ agers who are susceptible to organ-specific disease. And lastly, we believe that advanced plasma proteomics opens the possibility for really unprecedented insight into human cell and organ physiology on an individual basis, and most importantly, before people have a disease that manifests clinically. And I would like to thank our collaborators, both outside and at Stanford, my lab, and then these funding sources. Thank you very much.
Thanks, Tony. Lots to talk about there. I'm going to start with a question you probably got a million times and get frustrated with, but I get asked all the time. I'll answer it. So people will say to you, well, you take young blood from an old animal, you're diluting the factors in the old animal, and you take old blood from put it in a young animal, there's a lot of inflammatory factors. I'm sure there's more going on than that, but how much does inflammation and dilution explain phenotypes?
Yeah, I think we have to be somewhat cautious with transferring something from an old organism into a young and explaining what happens, because it's possible, as you said, that they're simply sort of, detrimental inflammatory processes. What's sort of a counter argument, and actually also for the simple dilution effects, is that when you use these unbiased single-cell transcriptomic approaches, you see signatures that are identical to what you see with normal aging. So let's say if a gene goes up with age, and then you expose it to young plasma, it goes back down to an earlier state. So yes, you could say, well, this might just be a dilution effect, but then I think some of the effects are clearly driven, like increased stem cell activity, by the presence of trophic factors and growth factors, that it would be very hard to explain that by having fewer of those factors, you would all of a sudden increase stem cell activity. I mean, nobody has shown that you can take, stem cells and inject saline and then you activate stem cells, right?
Yeah.
But at the same time, I think for some of the beneficial effects, a dilution is probably useful.
You kind of ended that portion of the talk with we need to figure out what the factors are that are in young animals that are having such a beneficial effect in old animals. And then you went to the proteomics. And when you look at the proteomics, there's so many things there, and many of which are changing. And that's only the proteins. There's all kinds of other molecules that are changing
Right.
as well. How do you sort it out and figure out where the signal's coming from?
Yeah, this has been the biggest frustration, and you're absolutely right. The discoveries from Tom Rando, were now more than 10 years ago, and so was our studies on the brain, and only a handful of factors, have been identified. And I think it shows you really the problem I think that we have in general in physiological studies, and particularly in respect to aging, that you have to conduct these studies in the whole organism. There's no cell models that can predict
Yeah.
what is going to be effective and what not. And the idea that you could do CRISPR screens in vivo is a great one, but it has been very challenging to do.
Yeah.
But this
Yeah.
is where we're going. So we believe the field has advanced enough, and single cell transcriptomics, but even more now single cell or spatial transcriptomics and proteomics allow you to really look at least 100 genes at the same time in a specific organ. But even there, you have to focus on one organ. You can do whole genome knockouts at
Yeah.
scale yeah.
I also think that we build clocks. We've built them from methylation, but we like building them from proteome or metabolome or lipidome because you're dealing with functional molecules there. But when you're
Right.
looking at things in the blood, I mean, the challenge is that you're seeing levels of some protein change in the blood, but as you say, that protein originated in some tissue. So what is the correlation? I mean, it obviously is going to vary from protein to protein, but in general, is there a correlation between the levels of a protein that's in the blood and the amount that's being produced in the tissue of origin at the time?
It depends, as everything in biology. But as I said, we use this in the clinic, right? We make predictions on organ function based on measuring, transaminases from the liver. And, they're obviously good predictors.
Sure.
You don't necessarily have to correlate um, completely, with a correlation factor of 1. Even if you have
Sure.
a lower correlation, if you then use multiple different factors, you can estimate the physiological state of an organ or a cell type even. Um, but yeah, it's a good point. I mean, we just recently actually, it just presented this yesterday at this conference here. So we have about 2,000 samples, paired samples of CSF and plasma from people with and
Mhm.
without disease. And that allows you to ask exactly that question. What is the
I was going
correlation?
to ask that question too.
Yeah. What is the correlation between proteins in the spinal fluid and in the blood? And overall, the correlations are really bad. They are better for proteins that are presumably produced in the periphery and make it into the brain than those that are made in the brain based on gene expression and make it into the blood. But there's some where you have very strong correlations, like leptin. And we use actually this information to make predictions about transporters or change in transporters. But it could also give you information about the stability of proteins in the different tissues and how that changes with age. But in the end, I think I see it more as a tool to get at important biology, and the biomarkers will be probably some sort of a composite of biology that we can't really explain. Like, some of the ratios we use in Alzheimer's disease, we have always used the ratio between A beta and tau. That has no biological meaning,
Yeah, yeah.
but it's an extremely good predictor of the state of the disease, right? So I think we can do similar things with a proteomic approach, but you're absolutely right. If we could, we would take a biopsy of each tissue.
Well, I'm not volunteering for that, so. Um,
Especially the brain one.
yeah, especially that, although that might be not big loss for me these days. Um, One of the big questions in the biomarker field is sort of systemic aging, measuring systemic aging versus organ specific aging. Are we just a compilation of degeneration of organs at different rates, or is there an intrinsic process that, a controller that is interacting with each organ to generate aging at different rates in organs? And you can sort of address that because you're measuring a sort of a systemic age and organ-specific age. So, what are the correlations there? Which do you think are better predictors? Are specific organs more aligned with systemic age than others?
Yeah, I don't think we have enough data to, even though we have 50,000 individuals, I think, this is just the beginning. I would like to see longitudinal data from the
Sure.
same individuals,
Yeah.
which, we have a little bit of data and seems overall, if you have an older heart, it stays an older heart. So it's not that you catch up after 5 years, although you might. And some of this is then the question, is it a true effect or not, right? Are what we're measuring is too noisy, or biologically actually your organ age varies as you get older, or throughout lifespan. Um, I think it will be extremely interesting to try to model this in animals. And we have a study with 1,000 mice and 9 genetic backgrounds where we have, together with Nadia Rosenthal at Jackson Labs, where we have plasma, and we're now trying to get funds to, measure the proteomes of these mice. And then we have all major tissues where we want to do single cell transcriptomics. And we have also functional data that Jackson has done and clinical chemistry. So in mice, obviously, you can control things much better, and hopefully we will get more insight into your specific questions. Is it, first of all, across lifespan, do organs influence each other? So if let's say you have an old heart, does that then lead to an old kidney and an old lung? Is there sort of a systematic way how aging could spread? And Carlos Belmonte is doing some of these studies where he accelerates age or he rejuvenates tissues, and tries to see how is the effect across different other tissues. And there's other approaches to try to do that. But I think it will be very interesting to explore this and understand sort of the spreading or the infectious nature of aging.
Yeah, I think so too. I think there's a lot to be learned. And I want to bring in Ching Zhang, she's a graduate student at the NUS School of Medicine, and she's going to be asking some questions from the audience.
Yeah, hi, Professor Risi. So there are lots of questions, interesting questions from the audiences. So the first one I may want to ask is which is better, is the serum or plasma in the therapeutic potential as a treatment for cognitive impairment?
Yeah, so the difference between serum and plasma is that um, serum is derived from letting the blood sit at room temperature for an hour, and then it triggers coagulation cascade complement, and all the immune cells basically go nuts. So it's a reflection of the potential activation of proteins and release of protein factors from blood cells. It's less useful to look at the physiological state. So I think you want to look at plasma because it's a reflection, presumably a reflection of the protein concentrations from the living individual. And the same is the case if you want to treat somebody. You definitely don't want to infuse them with serum because that would create a lot of problems and would actually activate the sort of innate immune system in the recipient. So you always get plasma in the hospital.
Yeah, true. So next question will be how long does the effect of the young blood will last?
Yeah, so in mice, Vadim Gladyshev and James White did a really nice study where they used a model parabiosis where you suture mice together. And they did this, and so then the blood circulation joins, and you have an exchange of blood from one animal to the other. And what they did is they used this model and left mice together for 3 months, and then they surgically separated them again. to ask the question exactly that you're asking, how long do the effects last? And what they found is they actually did a survival study of the mice, and they found that old mice that were paired with young mice lived on average, I think, 8 to 10 weeks longer than old mice that were paired with old mice. So clearly the effect does not go away, but lasts for months, for probably for some mice, up to a year. So that would be in human years, it would be, decades. They could also show epigenetic effects that remained for months and months. So it's not that it snaps back to the previous age of the mouse, but they're really long-lasting effects that make sense in what we have learned from these models. If you increase stem cell activity, you have more stem cells, you have more progenitors. They're not going to die if you now take the young plasma away, but they will have a long-lasting effect.
Yeah, that sounds quite long already. Yeah. And the next question will be, is it possible that the mitochondria transplantation is responsible for the rejuvenating effects of the young blood and the lipid?
Yeah, no, I think that's not really possible, because first of all, in the parabiosis model, you could argue that, the cells that are the blood cells that go from one animal to another somehow transmit their mitochondria. But with the models that we have used and others where you simply transfuse with plasma, we can actually filter that plasma, and there's no mitochondria in that plasma.
Yeah, then I think maybe for this part, that no mitochondria effects is included. And the next question will be,
But maybe if I may add, but in the unbiased analysis, in our analysis, and also from Guanghui Liu, mitochondria are a major target for rejuvenating and age accelerating effects.
yes,
So
there
there
are
are
new.
factors that target mitochondrial physiology, biology, but it's not through transfer of mitochondria. Sorry.
I see. So the next question will be, do people with healthy lifestyles have like younger looking proteins in their blood? Like, have you compared people with healthy lifestyles and then like
Healthy.
healthy lifestyles, like
Um,
exercises?
yeah, so exercise is definitely a beneficial factor, and we can show, and Solvi Leda also showed, that if you exercise mice, you can transfer beneficial factors in plasma and mimic the effects of exercise, at least on the brain. So that suggests again that there are circulatory factors that are responsible for some of these physiological effects. I'm not sure that answers the question.
Hmm. I think the audience is asking, like, have you tested like the proteins in the blood of maybe the mice, or maybe like clinical data, some clinical data of people with healthy lifestyles? Have you also like seen that all the signature proteins in the data?
Yes, we have done some of these experiments, yeah, and seen similar signatures.
Yeah, I see. Then the next question will be, is there any evidence that getting blood from younger family members, like Brian Johnson did, is better than from unrelated young donors?
Yeah, I mean, let's not talk about this person, I think.
Yeah, okay, yeah.
But in general, there the plasma that is being collected by companies for medical purposes, they pool it, and so The company we've worked with, Grifols, they collect tens of thousands of donations every week, and they pool them. Their average donor age is about 30. And once you remove the blood cells, there are no incompatibilities based on tissue. And so it doesn't really matter who the donor is. Now, there is a study in Norway where they use young athletes, to donate plasma. So because they want the best possible plasma. And they argue that, people who would exercise have even better plasma than people who don't. And that's probably true. So yes, it's true that it depends from who you get the plasma, but it doesn't have to be a family member.
Yeah.
Maybe one last burning question.
Okay. Then maybe given that the young blood appears to rejuvenate aging brain, so what do you see as the biggest hurdles in translating these findings into the safe and effective therapies for humans, especially for conditions like AZD, the Alzheimer's disease?
Yeah, that's a great question. I think it relates to Brian's earlier question, finding out what are the key factors and then making drugs from them. I think plasma infusions could potentially have beneficial effects, and there's some studies, even phase 2b/3 studies from Grifols, that show people have cognitive benefits, even in placebo-controlled studies. But I think to really make this available for everyone, we need to make a drug that is cheap and has sort of the main ingredients. And figuring out what the targets is going to be the biggest problem, I think.
So thanks a lot, Ching, and thanks, Tony. We don't want to keep you too long. You have to go enjoy Tuscany. So we'll have you back sometime in the future. Thanks a lot.
Yeah, thank you so much. Take care.
Thank
Bye.
you.
Too. I want to remind people to use the chat function and choose panelists and all attendees option to lead comments and feedback for everyone to see. The next webinar will be June 5th. It will be Yifan Yang. And we don't have his topic yet, so we're all going to be surprised, but I'm sure it'll be interesting. And then finally, to close out today's session, we're going to have a lighthearted moment from the TV series Abbott Elementary, where in honor of the first 100 days of school, the students dress up as centenarians. Let's see what they think centenarians look like.
Good morning, old ones. Yes, all of you centenarians. Well, be careful of that back now.
My arthritis is acting up.
Your arthritis is... Oh, I love your moo-moo. Now that is an appropriate use of a moo-moo on a 100-year-old.
Thank you, Mrs. Howard.
You're welcome. Oh, little Miss Margaret. Girl, you look fabulous.
Thank you. I'm you.
You who?
I'm dressed as you.
Well, why would you be dressed as me?
Because you're 100 years old.
I am not 100 years old.
But you're the oldest person we know. Say you're grandma.
Okay, first of all, the oldest person you know is Mr. Johnson. And secondly, grandmothers can be young.
You're way older than my grandma. You're even older than Mrs. Teagues, and she's like 50.
Miss Teagues is neither 50 nor a grandmother.
Mrs. Howard,
Yes.
don't be sad. You look great for 100.
Sure do, boy. You gotta tell me your secret. I don't look that good when I'm your age.
I'm feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down.